lab studies neutrophils in inflammation and a process particularly we call net formation or netosis. So you can imagine in your body a type of white blood cells we call neutrophils. When they are activated, they are just like Spider-Man. They will shoot out their chromatin and DNA content like Spider-Man shoot out the webs from their hands. So this is a process actually originally called a means of self or host defense, but later on a lot of clinical evidence show that this kind of nets that we call, they're generated in sterile inflammation where no infection is involved. They're very injurious to the body, they are pro-thrombotic, pro-inflammatory. So my lab is particularly interested to look into this net formation when we are looking into the context of diabetes and metabolic disease. So when I was in the States, I found that actually the net production is exacerbated in diabetes. And this is also linking to like a diabetic complication like delayed wound healing. This result has been actually corroborated by a number of groups in the world, and then soon afterwards people also identified the role of NETs in other diabetic complications. But the thing is that it's always a missing gap in understanding how these neutrophils are predisposed to generate more NETs in diabetes. So actually this is one of the important missing gaps also with unmet clinical needs, because if we don't know how this process is being generated or mediated, it's very difficult for us to intercept the pathway. So what we are doing in this lab now particularly is we try to understand the process and also the impact of this net generation in metabolic disorders. So my research program actually have 3 arms. So one arm is cell biology. We want to look into how these nets are generated, the pathways, the molecular entities that's involved. The second one is we use in vivo animal models to see, and also human samples actually, to see what are the impacts of nets on the in vivo system itself, and also the relevance to humans and their role in it. Can we intercept it to ameliorate some disease? The third one is actually net and bacteria. They can interact with each other. In ourselves, we already have a lot of bacteria. So one of the main things we're also hoping to do right now is to see how neutrophils and also our microbiota interact and how their interplay bring about metabolic disorders. So as you will see, actually this research program will allow target discovery, and at some point, as we know that neutrophils are being recruited at very early stage of disease, so it might also be instrumental for it to act as a biomarker. We're trying to evaluate the value and the possibility for NET to be serving as an early biomarker to detect like disease at early stage before they evolve into a bigger problem. Our overarching research goal is to harness what we know about in intervening pathological netosis to prevent or combat chronic inflammation. So intercepting chronic inflammation is actually very important because nowadays, when longevity is increasing globally, we want people to age healthily and functionally, not just as a number in increase. So our research, which encompasses cell biology, preclinical and animal model testing, actually allows us to make breakthroughs in target discovery and sometimes drug repurposing for inflammatory diseases. We also actively collaborate with clinicians and colleagues in engineering, so we try to develop pathways to hopefully bring our batch science discoveries to bedside application. Of course, all research starts with curiosity and continues with passion and determination. So I hope my lab also serves as a platform for junior researchers to develop their interest, to equip with knowledge, know-how, and qualities to be sustainable scientists. Nurturing new generations of scientists is one of my endeavors working in the medical school. So anti-NET treatment actually represents an excellent therapeutic approach to curtail NET-mediated ailments. So as of now, DNase-1 is the only FDA-approved anti-NET treatment available. But actually, these DNAs, they can only break down when NETs are formed. So you can imagine the bad or the injury is there already. So what we want is actually to prevent this dangerous or injurious substance or the NET released from the neutrophil. So what we want is really to inhibit the production of NET as the most ideal treatment of anti-NET treatment. So for us, because as you see, our research actually could shed light on new possible targets, and we hopefully would develop anti-NET inhibitors that will be instrumental to diabetic complications and beyond. As you can see, actually NETs involved in a number of disorders, including thrombosis, cancer growth, metastasis, even autoimmune disease, just to name a few. So we would be envisioning that actually the anti-NET treatment is useful for diseases beyond diabetic complications. It's actually a very exciting field to work in. I actually feel privileged to join LKCMedicine when it is still young. There are so much room for the school and myself to grow, to develop and to advance. The research community in LKCMedicine is also very vibrant and energetic. You can easily form collaboration even when we are just having a small casual chat. What's more important is LKCMedicine has strong partnerships with National Healthcare Group as well as College of Engineering in NTU. As you will see, for us to bring our bench science findings to bedside, these are very crucial elements to allow our research to reach the goals and impacts that we envision.